So here we're going to look at a very important type of molecule for the human body called an amino acid. Now, amino acids are the building blocks for proteins. They are the monomers for the longer polymer molecules that we call proteins. And we're going to look in more detail at these polymers in another video. However, it's important to note that, yes, these amino acids are the building blocks for the proteins in our bodies. Now, most of the amino acids that we deal with, as you can guess, amino acids are molecules with an amino functional group and a carboxyl functional group, making them amino acids. Now, pretty much all of the amino acids that we deal with in the human body are what can be referred to as alpha amino acids, or alternatively, they can be called 2 amino acids. And what these names both mean, they both mean the same thing. Basically just means that in the same way that we have the traditional naming system for organic molecules, this just means that the amino group in our amino acid, or in alpha amino acids, or 2 amino acids, the amino group is on the second carbon. So we know that when we have a carboxyl functional group on an organic molecule, we start the numbering from the carbon in the carboxyl functional group, making this carbon number 1 and this one here carbon number 2. And so in alpha amino acids or 2 amino acids, the amino group is always on this carbon here, carbon number 2. And then, so this is the general structure here for a 2 amino acid. We have our carboxyl functional group bonded to another carbon with an amino group coming off it. And then this thing here, which is often referred to as the Z group. Now the Z group can be any sort of chemical structure. It's not necessarily traditional functional groups, but it's any chemical structure of functional groups. So it's very rarely going to be a standard, straightforward sort of setup like we're used to dealing with organic molecules. And it's often the Z groups often get quite complex, and thus, because all of our alpha amino acids contain the carboxyl group and the amino group in the same way, with the amino group on the second carbon, it is this Z group here that really influences the characteristics and the properties of a particular amino acid. So all of our amino acids, as I said, have most of the amino acids in our body have the amino group on the second carbon with this Z group on the side. And now, because they all have an amino group and a carboxyl group, that makes our amino acids polar. So the NH2 group, the amino group, and the carboxyl functional group make them polar. And what that means is that they are, in fact, water soluble. Because of their polarity, they can form dipole dipole bonds with water molecules, and thus dissolve in water. So we've got all these amino acids that are obviously very crucial to our body and our body's processes. Now, there are a specific type of amino acids which we refer to as essential amino acids, which are slightly different. Most of our amino acids that we need, that our body needs, can be produced inside our body. However, there is a small number of amino acids that our body can't produce, and therefore we have to consume these special amino acids in our diet in some way, and these are what we call essential amino acids. So these are the ones that have to be consumed in our diets, because our body can't produce them. So these are kind of a few important factors, a few important sort of properties and ideas that relate to all of our amino acids. However, if we want to take a little bit closer at the actual chemical structure of the amino acid, then we'll see something very interesting. Obviously, because we are dealing with an acid due to this carboxyl functional group, any amino acid can act as an acid by losing this hydrogen atom here. And thus what we get is a negatively charged ion. Alternatively, however, because we also have an amino group here, what can happen is an amino acid can act as a base and receive another hydrogen atom in this amino group here. And what happens then is we have a positive ion because of the extra proton, the extra hydrogen nucleus that is joining on to this nitrogen here. So that is how an amino acid can also become a positive ion. Now that's very interesting. So our amino acids can act as acids by losing or donating a hydrogen atom from the carboxyl functional group, or they can act as bases by accepting a hydrogen from somewhere else and bonding that hydrogen with the nitrogen atom here, making them a positive ion. So they can act as acids and bases, and it is actually possible, in fact, for an amino acid to act as both an acid and a base at the same time. It can donate this hydrogen from the carboxyl group and accept a hydrogen into this amino group at the same time. And so up one end we have a negative charge, and up the other end we have a positive charge. Now there's a special term for this type of molecule here that can act as an acid and a base, or alternatively can act as a negative ion and a positive ion at the same time. And what we do is we call that a Zwitter ion. So a Zwitter ion refers to an ion that is kind of both a negative and a positive ion at the same time. If we got rid of this hydrogen here, this would be a negative ion. If we put the hydrogen back on this end, then we would have a positive ion. However, both ends of this amino acid molecule have reacted in either as an acid or as a base, and thus we have a Zwitter ion. Alternatively, we can refer to this as a dipolar ion. So when it's in this Zwitter ion state, an amino acid molecule has no net charge. It's up one end it looks to be a negatively charged ion, and up the other end it looks to be a positively charged ion. However, if we look at the molecule as a whole, the opposing charges cancel out, and the molecule as a whole has no net charge. So what happens then is if we have our amino acid, so if we go back to our original amino acid structure, then we can predict how it will behave depending on whether or not we're in basic or acidic conditions. If we're in very acidic conditions, it means there are lots of hydrogens floating around, lots of hydrogen protons. That is the nature of an acidic solution. And so that means if we're in an acidic solution, obviously there are lots of hydrogens around, which means one of them is likely to bond here. So that means that an amino acid will act as a base when it's in acidic conditions. Obviously vice versa, if we're in a basic conditions, then there are very little hydrogen atoms floating around in our basic solution, and that means the amino acid is very likely to donate its hydrogen atom into the solution so that the solution has more hydrogens or hydrogen nuclei floating around. So in the same way, and vice versa, it will act as an acid in basic conditions. So that's very important. And what that means is the fact that it sort of opposes its conditions, the fact that it acts as a base in acidic conditions, means that it sort of nullifies the acidity of its environment. By absorbing some of the hydrogens from its acidic environment, it means that its environment becomes less acidic. In the same way, by donating hydrogens to a basic environment, then it means that that environment becomes less basic, and in that way it acts as a buffer. So what it does, what it means by being a buffer is that it counters changes in pH in just that way. It kind of absorbs some of the changes in pH, so that the net effect on a solution containing an amino acid is reduced when we add hydrogen or add hydroxide. So that is what it means to be a buffer. Now we're going to look at two brief examples here. Two brief examples of ways that we have to look at our amino acids. So amino acids are often slightly more complex versions of the basic organic molecules that we're used to dealing with, depending on the nature of our Z group. Our Z group can often get very complex and be difficult to deal with when we're using traditional ways of naming our organic molecules. However, sometimes our Z group is actually quite easy to analyze, and thus we can actually find a more traditional name for our amino acid. So, for example, here we're going to look at an amino acid called isoleucine. Isoleucine has the structure like this. So obviously whenever we're dealing with sort of biological chemistry and that kind of stuff, we're going to refer to this molecule as isoleucine. That's just what it's called. However, we can in fact give it a systematic name using the regular naming rules that we've learnt. So this is a bit of a messy drawing here of this structure. However, that is the structure of isoleucine. So this is usually referred to as isoleucine, but we're going to try and find a more natural name for it. Well, not a more natural name, a more systematic name for it. So obviously we've got a carboxyl group here, so that is carbon number 1. The longest chain of carbons is through here, and is a chain of carbons containing 5. So we know we're dealing with something with the stem pent in it. Now, the carboxyl functional group does have, or the carbon in the carboxyl functional group is always numbered 1, and therefore we again have a 2 amino acid with the amino group on the 2nd carbon. We also have a methyl group on the 3rd carbon. So what that means is that we've got 3 methyl, 2 amino, and then because it's a carboxylic acid, we have 3-methyl 2-amino pentanoic acid. That is the systematic name for isoleucine. And the reason we can do that is because the Z group, or the Z group of isoleucine, or this chunk up here, is quite easy to deal with. We know how to deal with systematic naming of this type of Z group. Now, another example that we're going to do is we'll just look at a molecule, another amino acid. We're going to look at cysteine. So this is the chemical structure of cysteine. So we've got, again, the stock standard stem that we have for all our amino acids, like this. And so we're going to look at how this will react. We're going to draw how the structure of this molecule will change in certain conditions. So that is the structure of cysteine. Now, first of all, we're going to look at how this will look, how this chemical structure of cysteine will change in an acidic solution. So we know that in an acidic solution there are lots of H pluses floating around. And we know that because cysteine is going to act as a buffer, like all amino acids do, it's going to counter that. And so thus, cysteine is going to act as a base. And what that means, we can figure out both because it's acting as a base and because there are so many hydrogens floating around. We're going to get an extra hydrogen bonding in here. And we're going to end up with a positive ion when cysteine is placed in acidic conditions. Now, if we wanted to look at cysteine in basic conditions, then we could work this out again quite straightforwardly. So we know that cysteine acts as a buffer, so it's going to act as an acid in basic conditions. Also, we can say that in basic conditions there are very little hydrogens floating around in the solution, which means that cysteine is very likely to donate a hydrogen to the solution. So it's going to lose this hydrogen here and become a positive ion. Now, there's also a very interesting case. If we put cysteine into a solution of precisely the right pH, very finely balanced at the right pH. So this should actually be obviously a negative charge here, not a positive charge. Now, if we get cysteine into exactly the right solution with a solution at exactly the right pH, then cysteine will act as a Zwitter ion and will have both accepted a proton up one end and donated a proton up the other end, and that is when we can refer to it as a Zwitter ion. So that is basically everything we need to know about amino acids, specifically about alpha amino acids.